Hey, this is Tiffany Martin with Medical Microbiology, and we are working our way through Chapter 16, Adaptive Immunity. And we've been talking about T cells. We talked about how they're structured, T cell receptors. We talked about MHC and antigen processing, and we talked about clonal deletion. So now we have our T cells, and they have moved into the circulatory and the lymphatic system. They will fully mature and differentiate a little bit more into cytotoxic T cells and helper T cells. And then we need to see how do they learn, how do they get recruited to do their job. So they're there, they're waiting for their soulmate antigen, and then they will kick into gear. So that's the process we're going to talk about now, and that's called T cell activation. So we're activating these T cells, and we're saying this pathogen you've been training for, or you've been designed for, is now present. What do you do now? So that's T cell activation. So let's go to the PowerPoints, and we'll see how that goes. Okay, so we've talked about T cells. You know that they have a T cell receptor. You know they've gone through clonal deletion, so we kicked out all the ones that don't work, and any of them that will attach to autoantigens. They will fully mature into cytotoxic T cells, which can also be called CD8 cells, and helper T cells, which can be called CD4 cells. And these have different jobs. Cytotoxic T cells are called cytotoxic because they produce a different protein called CD8, and their job, their job is to actually kill. And remember, this is cell-mediated immunity. T cells target cells that have a pathogen inside them. So cytotoxic T cells are going to be the ones doing the killing. Helper T cells are actually going to do, or they're going to help the T cells, and they also help B cells do that job. And the way they do that is they secrete some signals called cytokines. So that's what helper T cells do. Now, helper T cells are called helper T cells because they secrete a different protein called CD4. And I remember this is just my little trick. T helper cells, the T and then H, a lot of times you'll see a T followed by a lowercase h. To denote helper T cells. If I take this H and I turn it upside down, it looks like a 4 there. So I know that T helper cells secrete CD4. Cytotoxic T cells, you can just think of those as the other ones, or you can take this C in the cytotoxic and make it into circles to make your CD8. But T helper cells are also called CD4 cells. Take the H and flip it upside down, you have that. All right, so helper cells, they will actually differentiate into two types, helper 1 and helper 2. T helper 1 assists cytotoxic T cells. T helper 2 are going to work with B cells. Now, there is a third type of T cell, and we have learned about this one already. These are called regulatory T lymphocytes. Do you remember where they come from? That's right. They are the ones that went through clonal deletion, and they recognized self. And most of those were killed, apoptosis, programmed cell death, but they kept a few which differentiated into regulatory T lymphocytes, and somehow they work to prevent autoimmune diseases. Not exactly sure how all that happens. But that's what they do. All right, so those are our T cells. Now, I did say that T helper cells help by producing cytokines. We need to define what cytokines are. Cytokines are proteins. They get secreted. So a cell will make them as a response to some sort of signal or chemical. They will make these proteins called cytokines, secrete them out into the environment, and then other cells can bind those proteins, and that's how they talk to each other. So that's what cytokines are. They're proteins that get secreted. Other proteins have a receptor and can tell that they're there, and that lets them know what's going on in their environment. Kind of reminiscent to quorum sensing. Do you remember when we talked about that in bacteria cells secreting quorum sensing molecules, and other bacteria could have receptors and they would know how many bacteria is in the way. So this is a cell signaling network. Now, the cytokines that we're going to be talking about secreted by our helper T cells are called interleukins. The ones that are secreted by leukocytes talk to other leukocytes, and we call them interleukins. So that's what you'll see on the next thing. Okay, so cell-mediated immune responses. How does this happen? So we're going to start with T cells, figure out how they respond to intracellular pathogens, and then see what they are. Remember, they also they're looking for endogenous antigens. These are pathogens that have already invaded a cell, and not only do they look for pathogens, but they also look for cells that are not behaving correctly, such as cancer cells. So that's the job of these cytotoxic T cells. Okay, so these are the steps in activation. We're going to go through antigen presentation, helper T cells, clonal expansion, and self-stimulation. So let's go to the picture because that's more fun. So let me do a drawing first, and then we'll come back to this one. I have drawn, this is a lymph node here. So this is part of our lymphatic system. A lymph is moving, fluid is moving through, and I have a dendritic cell, a dendrite. From that, remember, dendritic cells are the cells that are part of our skin and mucous membranes, and they were phagocytic cells, took something in, chopped it up, eliminated by exocytosis, and saved a few pieces. So my red pieces is something that this dendritic cell found and wants to go show the adaptive immune system and say, hey, this is close. It's in our skin. It's in our mucous membranes. Is this bad? Do you need to respond? So it moved from the tissues where it was into the lymphatic system. It's moving through the lymph, and it's now found itself in a lymph node here. Okay, it is presenting these antigens on an MHC2 and an MHC1. Remember, it has MHC1 because all cells have MHC1, and it also have MHC2 because it is an antigen presenting cell. So it also is able to present the same antigens on an MHC2, and that's going to be important when we get to the T cells. Okay. Okay, so it's in the lymph node, and it has now presented it on an MHC, MHC1, and MHC2 cell. And in the lymph node, we have our T cells hanging out. These are the ones that have gone through clone deletion. They have differentiated into cytotoxic T cells and helper T cells. Remember, cytotoxic T cells are producing a protein called CD8. Helper T cells, flip that H over, is producing a protein called CD4 from that. Okay, so in this cell, it has this antigen presented on MHC, and lo and behold, it has found its soulmate T cytotoxic T cell. Again, it moves through the body quickly. Within 24 hours, this guy will locate this guy. This is the soulmate antigen. This cytotoxic T cell has this unique T cell receptor, and it binds to this antigen. Now, cytotoxic T cells, remember, have CD8. That CD8 needs to bind to MHC. Remember when we talked about cytotoxic T cells, not only do they bind the antigen, but they bind that MHC molecule. So this also has to happen. Now remember, this is CD8, and CD8 are capable of binding to MHC1. Okay, now T helper cell is also there. He also has found his soulmate antigen. He's also bound here. He has a CD4 protein, and CD4 binds to MHC2. So cytotoxic T cells will only bind to antigens presented on a MHC1. CD4 helper T cells will only bind to antigens presented on an MHC2. So that becomes important. How I remember that, 8 times 1, CD8, MHC1, that's 8. 4 times 2, CD4, MHC2, that's 8. 8 is your magic number, so you just need to match both of these up so that they add up to 8. So 8 times 1, 4 times 2, they all equal 8. Okay, so that's the only way that works. All right, so let's go to our PowerPoint and see what's happening on the PowerPoint. All right, so there is my dendritic cell that is in my lymph node. He has found his cytotoxic T cell. There's my MHC1 cytotoxic CD8, 8 times 1. CD8 is bound to MHC1 and has made a complex here. This is also called an immunological synapse or an immunological complex. This has to bind. Okay, so we have game now. This dendrite has found a pathogen, the CD8 that was designed to bind to it, this T cell receptor with its CD8, cytotoxic T cell receptor, is bound here. And so things are going to start, right? All right, we're going to wait at this point. Cytotoxic T cell has found the bad guy, but remember I said these T cells are the big guns. This is the huge defenses, and we don't want to unleash them unless we're absolutely sure. So this cytotoxic T cell thinks he has a bad guy, but he's going to wait for a minute. He is going to wait while T helper cell also identifies this. So the T helper cell also will bind. Remember, he's a CD4, so he's going to bind to MHC2, and he binds to this dendritic site. Now, he also has bound, and when that happens, there are some interleukins that are produced. I don't care that you know the number of them. There's a whole bunch of them. But just know that T helper cell, when he binds, remember that's his job is to help, and he helps by signaling. When he has identified, yes, this is a bad guy, he will start making interleukins. These will cause him to differentiate, and we're going to call him a T helper 1 cell, because he will start making some different interleukins, some signaling molecules. So T helper 1, because he is bound, will differentiate and start making some new interleukins. And so we're now going to call him a T helper 1 cell because he's making some new chemical signals. All right, so interleukin 2 is being made. Well, my cytotoxic T cell, remember he's waiting for some confirmation. This is the confirmation. These interleukins produced by this T helper 1 cell says, yes, I agree, this is a bad guy. Cytotoxic T cell has a receptor for these interleukins, and as soon as he starts producing them, they will bind to this receptor, and that's the go signal. That tells this cytotoxic T cell, it's time. You need to activate, you need to make an army. So he will now start clonal expansion. He will start dividing, he will start replicating, he will create identical cells to make up an army, and we call these activated T cells. They are ready to go. They've gotten the signal from the T helper cell. First they found their antigen. They waited for confirmation. T helper cell bound, found the antigen, agrees it's a bad guy, it's foreign, it's worthy of an immune response, created the go signal, these interleukins, receptors here, and now they have divided, replicated, and we now have an army. This is called clonal expansion. Now, one thing about this army is that he now will make his own interleukins, so he never needs help from the T helper cells anymore. He is able, because they agreed, he is able to activate and make his own interleukins and make sure that he can always respond against this particular bad guy, this particular pathogen. This is called self-stimulation on that. All right, the majority of the cells will be activated T cells. There will be a few that are classified as memory T cells. They don't immediately go out to kill cells. These are long-term cells. They can persist for weeks, months, years, maybe even decades, so that if you ever see this bad guy again, they can respond much quicker. They will never need a helper T cell anymore. They have agreed it's a bad guy, and he can respond much quicker and go right into clonal expansion and make an army. So where is this army going to go? All right, he is ready. He knows what he's looking for. He's looking for this antigen that started it all. And he's going to go out. It's a T cell, so he is looking for T cells, not T cells. It's a T cell. He is looking for any cell in the body that is presenting this antigen. So any cells that have been invaded by pathogens, that's what he's looking for. He's looking for those cells. They will have their MHC1 presenting that antigen. Remember we talked about MHC1, if a virus or anything invades, he's going to put a little piece of it and put up those MHC1 and say, hey, this guy's in me, come help me. That's what this army of activated T cells are going to do. Okay, so we know what these activated T cells are going to do. Memory cells, I have one more slide on that. Remember we said some of these activated T cells will become memory cells and they will stay around. And then the next time you see it, it can respond very quickly and without involving the T helper cells. So that's an advantage for that. This was considered T cell activation. This is probably one of the biggest concepts in this chapter. So go over it a few times and you're gonna have to talk this one out too. So find you a friend, find you a study group, and you guys go through it, bring your questions to Zoom, and we will do this in our Zoom as well.